Thank you for inviting me and thank you for tuning in. About 1 in 3 patients with mental disorders are resistant to current forms of treatment. And that, as you're well aware, is a fairly major issue. And so, what do we do about this? One of the challenge with providing treatments to the treatment-resistant people has been the fact that mental disorders typically affect deep brain circuits, including the limbic structures and the basal ganglia. And it has been quite difficult to modulate these deep brain structures and bring these circuits back to normal state. You may think that one approach could be deep brain stimulation, right? So that surgically implants leads of electrodes into specific targets and stimulates. Um, that has been worked quite well for motor disorders, but it has been quite difficult to apply this for mental disorders. The general notion there is that the risks of the surgeries are quite high. In terms of approval, there's only obsessive compulsive disorder so far as an option under humanitarian device exemption. So we really need non-invasive approaches where the risk is relatively low. And there are emerging electrical and magnetic approaches. Electroconvulsive therapy, transcranial magnetic stimulation. They can help people. One of the drawbacks that they have is that the fields are strongest at the source and fairly low in the deep brain regions that we ultimately would like to modulate. You're aware of ultrasonic waves having the remarkable property of being able to be focused at depth while also non-invasive, such as it is illustrated here. And there have been several emerging studies, especially in the recent decade, showing transient effects of low-intensity ultrasonic neuromodulation, such as shown here in this video, where ultrasound is coupled to the motor cortex of a rodent. And you see, following the brief pulse of ultrasound indicated by the LED, there's an immediate transient response. The response is transient, it goes away eventually. But if we think of the ultimate treatments, we would really want to reset the circuits, like rewire them, as is the case with electroconvulsive therapy currently. This applies strong currents into the brain, big portion of the brain, and there are often beneficial effects that are long-lasting. So in the same manner, we would like to apply ultrasound now with the main benefit that we can only modulate and rewire the circuits that we would like to, and sparing all other tissues and circuits, and hence mitigating side effects. We and others have learned that ultrasound modulates cells through activating ion channels, through its mechanical aspects, either the radiation force or the cycle-by-cycle displacement. And others have found that not only neurons, but also glial cells are being activated. There can be fairly complex cascade of events where the ultrasound activates mechanosensitive ion channels that can lead to increased intracellular calcium, and that can in turn activate other cell structures such as calcium-sensitive ion channels, and lead to action potentials and other events. As I mentioned before, ultrasound can also activate glial cells such as astrocytes here, and that there's a cascade here in a beautiful study showing that the ultrasound can ultimately lead to changes in synaptic activity through activating NMDA receptors. And the cascade starts with the activation of TRPA1 receptor and ends with the changes in synaptic activity and potentially also plasticity. So this is very promising, this mechanistic insight, and we would really like to test the potential of that in terms of treatments. So the hypothesis is that the longer the ultrasonic stimulation and the more energy we deliver, the more sustained and durable these neuroplastic effects will be. Of course, we cannot quite do it yet in humans. We cannot rewire someone's brain quite yet. And so we have chosen a close species to us, the macaque monkey here, to begin to test that potential. And so the monkeys have an ultrasonic transducer that is installed always into the same position. We have validated targeting inside MRI, and the animal is non-anesthetized, so we can engage them in behavioral tasks, such as visual discrimination, shown here. And by we, I mean my postdoc, Taylor, and my graduate student, Matt. So while the animal engages in that visual discrimination, simple looking left or right, let's say, a task based on some parameters of the stimulus, we focus the ultrasound into specific deep brain targets, such as the left lateral geniculate nucleus or the right lateral geniculate nucleus. These are MRI thermometry scans for validation of the targeting. And as a first starting stimulus, we are using just a 30-second train of pulses at 650 kilohertz, so relatively weak stimulus. Yet, we are already seeing robust effects on the animal's behavior. So in green, we apply the ultrasound for 30 seconds, so only 30 seconds, and you see that following this application into either the left lateral geniculate in some sessions or the right lateral geniculate on other sessions, we can actually shift the animal's preference for whether he's going to look left or right. And that shift is specific to the stimulated region. And quite remarkably, the effect flips polarity when you flip the stimulation hemisphere, left versus right LGN. And what was very intriguing to us that the effects actually stay or even build up over time and are observed up to maybe 20 seconds following this 30 seconds brief stimulation. So this is very promising. We also recorded neural activity from visual cortex using implanted electrodes, and we do see a clear change in the gamma activity following the ultrasound application. And the time course of that effect is does quite well correspond to the behavioral effects that we see. So this suggests that the LGN was really activated, and it was activated in a durable way. The ultrasound was turned off. There was no more stimulation, so it cannot be possibly an artifact. There are some durable neuroplastic changes taking place following the stimulation, which is very, very interesting. So this clearly opens the question, how long will these effects last when we increase the duration and the energy that we deliver into these deep brain targets? And that is something that we are investigating currently under the support of Focus Ultrasound Foundation. Just to tell you briefly, we didn't see any safety issues when we stimulated these deep brain structures. Day from day, the monkeys would work the next day. We didn't really observe any behavioral deficits. So let me summarize. We have a major issue in that about 1/3 of patients with mental and neurological disorders are resistant to drug treatments. Ultrasound has a great potential to non-invasively modulate the specific targets in a selective manner. But there's been a key barrier, and it has been the need for sustained or durable effects of the ultrasound so that we can apply the ultrasound and then the effects are beneficial for several weeks, let's say, in a given patient. And we are finding that so long as the ultrasound is of sufficient duration and energy, there are in fact durable effects that outlive the stimulation. Those certainly cannot be explained due to an artifact because they occur when the ultrasound is no more there. So this is very exciting, and we are now systematically increasing the stimulation energy. We haven't seen any safety issues, so we are moving forward with this with a great benefit, potential benefit to patients. This work has been funded by the Focus Ultrasound Foundation and by the NIH, and it has been performed by a set of remarkable students and postdocs in my lab, and I much appreciate your attention.